Today we're going to have an introduction to discussions about the eukaryotic microbes. And this is a whole group of organisms that, humans are certainly eukaryotic organisms, and so are the fungi, so are the algae. So we're going to talk about the eukaryotes in particular with regards to which ones are studied in microbiology. And that's important because half of microbiology is really all about the eukaryotic cells. So let's talk about which organisms that are eukaryotes are studied in microbiology. So these organisms include the fungi, the algae, the protozoans, and the helminths. So you might ask, well, what's a helminth? A helminth is a worm. Okay, so, and then, then you might ask, well, why are they called microorganisms if some of them we can actually see, right? And there are some worms that are actually 30 feet long. The tapeworms can grow that long, by the way. And so really it's just kind of a grandfathered type of discipline that started way back in the 1700s, even before we had microscopes. So and the last category of organisms are the parasites, and the parasites are not really classified as such because they're a combination of insects and protozoans and helminths, but they're classified because their livelihood is necessitated by living in or on another organism. So all throughout this part of the course, we will discuss various examples of parasitism in these other classes of organisms. So in order to really talk about eukaryotic cells, we need to talk about what is this cell theory. And the cell theory really is about the integral nature of the cellular unit, that the cellular unit is really the building block of tissue in the human organism. It's the building block of multicellularity. Some of the eukaryotic organisms we'll study only are one cell, and then some, of course, are multicellular. So really, the emergence of the eukaryotic biological design was groundbreaking because it enabled development of multicellularity. So in terms of studying the human body, for instance, it's very important to think of disease in relationship to the cells, because yes, the body gets diseased, but really it's all of the cells are the integral unit in which the disease influences. And when the cells get influenced, or when the cells become ill, then the body becomes ill. So we're going to, as we discuss some of these diseases in relationship to the systems they affect, we'll also talk about the cells. Now, the evolution of multicellularity was, as I explained, a groundbreaking design change, so to speak. And it really, because the prokaryotes were, they could colonize and they live in what's called these biofilms, but they don't segregate their functioning and become multicellular. So what I mean by that is that, at least in the human organism, we have various tissues, and in these tissues, all the cells, and in these organs, all the cells are very similar, and certain functions of those cells are enhanced, while other functions are regressed. So, as an example, the erythrocyte has no mitochondria, no nucleus, and its primary role is to carry things, to carry oxygen, carbon dioxide, and nutrients. And so, obviously the erythrocyte can't survive unless we have, bone marrow. We have to have bone tissue and places that the erythrocyte can get produced. And so what I'm trying to indicate here is that this multicellularity, part of that design change, was really to segregate out functions of whole organisms in terms of tissues and in terms of collections of cells. And that happened with the eukaryotes and not the prokaryotes. So in terms of this evolutionary process, it began quite many millions of years ago. In fact, as early as 2 billion years ago, these cells stopped just being independent, right? And they began to include organelles, and they began to diversify in terms of the different things that they could do. And in order for them to diversify, they had to take on these organelles that enabled them to expand the repertoire of not only what they could do, but also in terms of where they could live. So these pictures here are of the red algae, and they're fossils. They're 1.2 billion years old, and at least on the left here, we have this area here, which shows this kind of budding, this creation of more than one cell. And this was done to assist with attachment to the substrate. And then the other right-sided figure, you can see in this area here, this kind of divisions that were creating these cells into disc-like shapes. And that was the evolution of becoming multicellular as well. So they were, I guess they started out as these disc-shaped cells, and then eventually they evolved into red algae. So we'll talk about them in relationship to our discussion about algae. Now here is a timeline, and the way to read the timeline, you have to be, because I started reading it here. No, you have to start reading it here. So this is 4,500 million years ago, right? The Earth was formed, and then we went forward in time. So the important thing is that, of course, the bacteria, the prokaryotes, occurred before the eukaryotes, and we initially had anaerobic bacteria, and then we had the oxygen-producing photosynthetic cyanobacteria, and once that happened, then we could have aerobic bacteria. So then as we continued to evolve, then we had the appearance of the protists, the first primordial eukaryote, and then that protist began to take on, to engulf, so to speak, these organelles that were originally bacteria. And then at this point here, we had our red and green algae, and then we diversified. That's when we started developing multicellularity. So this is the process of what's called endosymbiosis, where we actually have our ancestral anaerobic, at this point, eukaryotic cell that took on these bacteria. And one of the bacteria, this is also groundbreaking, that was taken on was this aerobic bacterium. And so once we took on that aerobic bacterium, then we could begin to metabolize in the presence of oxygen. So here, then we, at some millions of years later, became a aerobic eukaryote. And then some of these eukaryotes actually took on these cyanobacteria that had the capacity to conduct photosynthesis. So we took on this photosynthetic cyanobacterium, and then eventually our eukaryote became a plant, right, with chloroplasts, and our non-photosynthetic eukaryote became our heterotroph, which is an organism that requires the energy from others to survive. And so that's the beginning of multicellularity, and certainly that has continued to be a very successful biological design. So back to our phylogeny, we have a separate lecture on this topic, by the way, but this is just a review. We have these 3 classes of organisms, 2 of which are prokaryotes, and then we have our eukaryotes, right? And our eukaryotes are what we're going to discuss in relationship to these microbiological organisms. Okay, so what is the organization of the eukaryotic cell? We have our nucleus, right? And so in biology in general, right, we start out with these biomolecules, and as we go in this direction, right, we become more complex. So we originally start with glucose and amino acids, and then we have cellulose, and in relationship to the plants utilize cellulose, which is a close cousin to glucose, in terms of building structures and cell walls. We have our amino acids and proteins that serve as protein channels. We also have lipids, and we form our plasma membrane that way. We have our nucleotides, our DNA, our chromosomes, and they are all housed in our nucleus. Now remember, the prokaryotic cells don't have nuclei. They have clustering of their DNA, but they don't have this extra double-layered membrane surrounding the nucleus. So here is our prototypical animal cell. Okay, and you can see that there are quite a number of organelles, and depending on the level of training that you're trying to achieve here, I'm going to go through all these different organelles, and then the way I teach my classes is really to bring out the organelles that are important in relationship to these organisms in terms of their pathogenicity, just like we talked about with bacteria. What is it about some of these protozoa that might make them more pathogenic, or more able to be recognized by the immune system, and all sorts of things like this, which are very important in relationship to microbiology. So here we have our nuclear envelope, right, and it segregates our chromatin, our chromosomes, from the cytoplasm. We have the double-layered, the lipid bilayer plasma membrane, which regulates movement of materials in and out of the cell. It also keeps a separation between the fluid in the cell and the fluid outside. We have mitochondria that are involved in oxidative metabolism and production of ATP. We have our rough endoplasmic reticulum, where we have protein synthesis occurring. And that endoplasmic reticulum is basically continuous with the nuclear membrane. So it makes it an easy transition and channel for the RNA, the messenger RNA, to come across and transcribe and translate the amino acids into protein. So then we have ribosomes, which is actually where the protein synthesis occurs. And then some of the other organelles include the perioxysomes, which destroy peroxide, the cytoskeleton, right, that's involved in a number of important functions, supporting the cell, providing a scaffold, infrastructure, and assisting in movement of the cell. So we have lysosomes that contain digestive enzymes. And then we have the Golgi apparatus, which is a transporting mechanism, and it packages and targets protein to export to the outside of the cell. And then, of course, our smooth endoplasmic reticulum, that is the site of lipid synthesis and drug metabolism. So this is basically the prototype eukaryotic animal cell. And again, we will discuss some of these organelles in relationship to infection and recognition and so forth. So our plant cells, now you're thinking, well, why are you studying plants in microbiology? Well, guess what? Algae is a microorganism, and algae also has photosynthesis. It's considered a plant. Okay, so we have a plasma membrane with our plants, but we also have a cell wall. Now, the cell wall is just like with the bacteria, right? It provides that extra protection because, well, the animals, right, the heterotrophs can move around, but the plants are stationary, and they need to be able to withstand dramatic changes in the environment. So we also have chloroplasts, and chloroplasts are the energy production machinery of the plant. That's where our green chloroplast pigment molecules reside, and that organelle engages in photosynthesis. We have starch granules that are places where these complex carbohydrates, after being produced by the chloroplast, get stored. We have the thylakoids, which are part of the ATP synthesis process. We also have a Golgi. We have a cytoskeleton. We have ribosomes. Plants do engage in protein synthesis. We have a nucleus. We have smooth endoplasmic reticulum. We have the nucleolus and the mitochondria. So you're thinking, well, you know, does the plant have a mitochondria? Yes, it does, because it engages in energy in oxidation and production of ATP in that regard. So our first example is, what are the differences between a plant cell and an animal cell? Well, number one is energy, right? That a plant has chloroplast, and an animal has a mitochondria, but so does the plant. The plant has a mitochondria as well. But, you know, more importantly, right, is this concept of autotroph and heterotroph. So the plants have the capacity to produce their own energy. They don't need to consume other organisms to, you know, get enough energy to survive. They produce their own energy through photosynthesis. Whereas you know, animals, so to speak, they're heterotrophic, and they require consumption of other organisms. So that's a really big difference. Another difference is the cell wall. Okay, plants have cell walls and animals do not. Now the third is not necessarily a difference between all organisms, but I'll just put it down here. So motility, right? So plants are relatively stationary, but I'll put a little asterisk here because some plants move. So the algae, of course, can be stationary, but it can also move. And then animals, of course, move. But, you know, potentially they can be stationary as well. What about the anemones, right? The various shellfish just, you know, stay in one place almost their whole life. So in that regard, they're stationary as well. So these are some of the major differences. There are little minor differences as well, but these are the most important differences. And, you know, again, we will be considering both plants and animals in our discussion about these microorganisms that are eukaryotic cells. So which eukaryotic organelles do you think were acquired from bacteria? Well, guess what? Probably the functions that the eukaryotes have that the prokaryotes do not have, right? So number one, mitochondria. We saw in the earlier slide the endosymbiotic process where that cyanobacteria that engaged in aerobic metabolism was taken up by the eukaryotic cell. The other is the chloroplast. We saw that as well. Okay, and probably some of the other organelles as well. Now, you know, the bacteria engage in protein synthesis, but the thing that the prokaryotes do not have is the nucleus. Now, the prokaryotes have that collection of DNA, but that DNA is not surrounded by a membrane. So somehow the nuclear membrane was taken up into that, primordial eukaryotic cell, and that change in design, so to speak, really made a huge difference in relationship to protection of DNA. Protection of DNA, which also then enabled development of more diversity. And more complexity in terms of genetic diversity. Okay. So that concludes our introductory lecture about eukaryotic microbes. Thank you so much for visiting educator.com.